So let's review everything we talked about so far. On the last video, we talked about the process of abiogenesis and how do you go from molecules of life to actually living things. And we talked about the fact that you start with monomers diffusing in and out of fatty acid cells. And once inside the fatty acid cells, some of these monomers may spontaneously polymerize and create extension templates and copy templates, which will then increase the amount of material inside of the vesicle. Heat from hydrothermal vents would also increase the polymerization rate, increase the permeability for monomers, and separate strands, increasing the amount of templates, increasing the rate of replication. The more polymers you have inside the vesicles, the bigger the osmolarity of those vesicles, and the more water goes inside. They get larger and they start absorbing the smaller vesicles. That means they eat each other up. Now that means the ones that eat each other up will become larger and larger and then tend to break into smaller chunks. Each chunk will have its own amount of polymers inside, but the polymers are self-replicating and they will repeat the cycle. Any polymer that replicates faster than others will become more common in the population, allowing the vesicles to grow larger, faster than the other ones, and split faster than the other ones do. Before you realize, these will become more common in the population and dominate the early population of little vesicles. And what you have there is essentially protobionts. So let us ask the question, does this already classify for the characteristics of life? Let's think about it. Does it perform metabolism? Well, it certainly does. It is making copies, it's extending the molecules, it's polymerizing, it's basically doing, anabolic reactions. Does it interact with the environment and respond to the environment? Yes, it's splitting up, it's absorbing things, it's interacting materials with the environment. Does it reproduce? Yes, because it's breaking to smaller pieces, and those pieces reproduce faster if they have mechanisms to do that. Does it evolve? Well, yes, because anything that gives an advantage to make this replicate faster will take over the population and become more common over many generations. Is it already containing maybe organization, order, genetic material, things like that? Well, maybe these sequences don't have any specific code. They're random sequences. But just the fact that they're there does provide an advantage for the organism. And very soon you're going to see the evolution of a genetic code. We'll talk about that in another video. But most of the characteristics of life are already being satisfied at this stage. This is life. This is what we call a protobiont, the earliest life. But how do you go from that to the life that we have today? So let's think about it. Which vesicle is going to do better than all the other ones? Well, remember, if the molecules self-replicate faster, if the lipids are a little stronger or better, if the structure is a little more organized, any of these things will give advantage to the vesicles, making them become larger and eat up the smaller ones, and then split up and repeat the cycle. So anything that focus on faster replication will give an advantage to the vesicles. So let's think about some of the things which will allow that to happen. Any vesicle that contain a polymer that only use whatever polymers were common on the primordial soup would have an advantage, because it would become more easier for that polymer to replicate. If you have a rare polymer in your sequence, that means that it's going to be harder for you to replicate, because you won't find on the primordial soup what you need to replicate. And because of that, very soon you're going to lock into those 4 common nucleotides, because they're going to be the ones most common in the primordial soup. And very soon they're going to outcompete any other vesicle that did not use the common nucleotides from the beginning. Any structure that makes replication more difficult by making weird folds, which makes the replication process more difficult, will be selected against. Any structure that actually makes replication easier will be selected forward. And that's what you see in the second picture. Any molecule that forms secondary structures that gives the molecules enzymatic properties which help accelerate chemical reactions will be selected against. In other words, early enzyme-like things. Catalytic molecules would have advantages because it will speed up chemical reactions. Remember that. Any sequence which will fulfill multiple purposes, like, for example, ribozymes, RNA, which can act like a structural role, which can act as an information transfer and storage, which can act as templates and enzymatic reactions, which can act as a transfer molecule. So RNA molecules we perform multiple roles will get an advantage over other ones. Any molecule that helps speed up the rate of replication will also have more advantage than the other ones, and we selected for. Any molecule that tends to split very easily and act as a good template for replication will be selected for. Any molecule that helps increases the replication rate would also be selected for, and will soon become more common in the population and be the most alright. Any molecule that help charge other molecules to already provide the energy so it didn't rely on heat, and that molecule can be charged so that polymerization can be more spontaneous and more quick, would also give an advantage to the vesicle, and therefore that would become more common in the population. Any vesicle that contains sequences which allowed the synthesis of lipids to make the membranes more efficient, or to increase the formation of nucleotides, or increase the formation of other molecules, would also become more common. Any molecule that help integrate with the membrane and enhance the interaction with the membrane to the outside environment will also become more common. Any molecule that increase the regulatory pathways so that the cells did not waste energy doing things that are not going to help us survive would also increase selection. Any molecule that help the cells communicate with the exterior environment, cell communication, will become selection. Any molecule that perform motor tests within the cell, carrying things from one place to the other, will increase selection. Any molecule that increase the fidelity of the copy process, making sure that the sequences were maintained the way they're supposed to maintain, will be selected for. Any molecule that sped up the copy process will be selected for. Any molecule that sped up the process of creating other molecules will be selected for. Any molecule that was more complex but that it gave an advantage to the cell will be selected for. And before you realize, you're going to have these replication and fidelity systems that can perform multiple functions, and we're going to get closer and closer to what we have today. Now it's just natural selection. It is not random anymore, because the sequences, they used to be random and used to provide an advantage just because their presence was making the cells growing faster by osmolarity. Now the sequences will start to become specific, because any sequence which provides any advantage whatsoever will make that become more common. And any of the advantages which I already described will make the thing become more common in the population. So what we're talking about here is about a process that's based on physics, chemistry, biology, thermodynamics, selection, and basic principles of science, all of which have been observed in isolation, in small steps. And even though we haven't observed the whole process from scratch, we do understand that it is possible for this to be the origin of life, because it just makes sense. Now, one thing that is for sure is that the very first molecules that were trapped on the inside and had advantages over the other ones were early RNA-like molecules, because the RNA is a very versatile molecule. It can act as a template to itself to be copied. It can act like a molecule that all you need to go from RNA to DNA is basically make a double-stranded RNA and then switch some of the bases, where it's from uracil to thymine, and now you have DNA. And then once you have DNA, that's one gene. But if you get that and attach itself to another gene, now you have more integrity, two genes sitting together and traveling together. So now you're starting getting longer and longer chains, DNA-wise. RNA can also double as an enzyme. It can act as a structural molecule. It can act as a transfer molecule. It can act as a carrier molecule in some cases. So because RNA is such versatile and does so many jobs within the cell, evidence suggests that it was probably the original molecule of life, and that DNA will come from that, and that proteins will come from that, and that the process of the DNA dogma, which is what DNA codes for RNA, which codes for protein, developed later because it created more specificity. And we'll talk about that on the next lecture series, when we're going to talk about where does DNA code and the process of DNA copy process and transcription and translation all come from. So I'll see you guys then, and until now, I hope you understand the process of abiogenesis. I'll see you in the next video.